Okay, now we're on to lab report format. All right, this is a hypothetical lab situation, and because this is distance learning, I can't make you guys do any labs. All right, but imagine if you were trying to blow bubbles, and there were two chemicals that is supposed to affect bubble size. All right, so I'm gonna go over your lab report format just in case using this hypothetical situation. Okay. Okay, so first off, you want to start off with the introduction of your lab report. Okay, this is again lab report format, how you will document science. Okay, you want to first start off with some background information. Okay, over here, because of the hypothetical situation of what are soap bubbles. If you guys are finding a hard time on what to write, what I usually do is go over the five W's: who, what, when, where, why. Obviously, you cannot use all five, but it's just, something to kind of go over. If you can use it, then use it. For here, for example, you can't use who for soap bubbles, obviously. So you may want to skip that one. Okay. All right. Once you guys are done with your background information, you're going to want to write out your research question. And this is because you guys are in high school in a actual research. This may be drafted out in a paragraph. Okay, now below your research question, you guys will have the answer to that, your hypothesis. Of course, at this point in time, you guys have already done your experiment, so you have already written your alternate and null. Do remember, we have to write the null hypothesis first, the H0, because the null hypothesis signifies that there is no change. All right. If this one is accepted, there is no significance of this research to the world. Okay, and in that case, it is the most unbiased statement. So you always write your null hypothesis first. Okay, next you guys have your alternate hypothesis. All right, that's the hypothesis that does show a change or there is a correlation in the data. Okay, your alternate hypothesis is also the same hypothesis you've been creating ever since your first science class. Okay. Okay, next will be your materials and methods section. All right. In a real research paper, this will be paragraphed out. If I was to ask you to write a lab report, this would be listed so it's easier for the teacher to grade. Now, how concise does the materials and methods have to be? It has to be, here, take this analogy. If you were to write your materials and methods section, it has to be so concise that a freshman reading it can recreate your entire research. Okay? Hope that's clear enough for you guys. Okay, now we're on to data. All right, data. Now, the raw data, the numbers that you collected from your experiment, that will be your quantitative data. Okay, the numbers. Okay, the numbers, that measurements that you will be putting through analysis tools. All right, will be your quantitative data. All right. Next, you also have what's called qualitative data. Qualitative data is used to emphasize points. It's used to describe. It's not as important as your quantitative data. All right. Your quantitative data is the important stuff here. Okay. Okay. Now, your quantitative data will be run through certain analysis tools. All right. In real research, here are the main three. These three are used a lot, especially in biology. All right, you've got the ANOVA. Analysis of variance is what the ANOVA stands for. And basically what it analyzes is whether your data sets are different enough to be significant. Okay? And if they are not different enough to be significant, then of course you would have to accept the null hypothesis. All right. If there is enough significance, in the differences between the data sets, then you guys would accept the alternate hypothesis. Okay? Now again, it's a analysis of variance. It sees whether the multiple data sets, whether they are different enough between each other, or are the natural variances within one data set, all right, whether the natural variances are larger, right? If the natural variances are larger or more significant, then obviously, of course, the data sets are not different. Okay, so that's what the ANOVA, or the analysis of variance, tests for, whether the different sets of data are different enough compared to the natural variation within the set. Okay, I hope that didn't confuse you. I do like to use ANOVA a lot, especially in my honors class. All right. Okay, next is a chi-square distribution. Okay, your chi-square measures the difference between the expected results or calculated results and the actual observed results. Okay, in chemistry, if you guys were to do research in chemistry, they do use this one a lot, the chi-square. It's also used a lot in ecology or environmental studies as well. Okay. Okay, last one, the t-test distribution. This is comparing the mean, all right, the sums and the means between two groups. Okay, is the means between the two groups different, significantly different enough, or are they not? All right, that's what the t-test distribution is used for. Okay, and then of course there are a huge amount of other statistical analysis tools, like your mean, median, mode, all right, that are used frequently. Okay, now keep in mind that if you guys were to use these three, all right, these three are the ones that will tell you whether you accept your alternate hypothesis or your null. They'll tell you whether it's significant enough, whether the the change or the data is significant enough to accept the alternate. Therefore, if you're using one of these, you want to set up your hypothesis to match it. All right. So the ANOVA measures a difference of variance between multiple data sets. All right. So you would use a hypothesis kind of like is something affected or not. All right. Chi-square, expected results and observed results. Okay. So your hypothesis would be something like is the calculated the same or different from the observed? Okay. T-test, you're just comparing two. Okay, so this would be the easiest in creating a hypothesis. So if we're going back to the bubble difference, does sucrose change the bubble? Same is the sucrose solute bubble solutions the same as the sodium chloride bubble solution. All right. That would be, if it's same, that would be the null, and then the alternate would be something like the sucrose solution is different from the sodium chloride solution. Okay, so you guys would compare the means and make sure you set up your hypothesis the same way. All right. Okay, as for the mean, median, and mode, I don't think I need to go over those with you guys. You guys should be pretty good at that. Okay, lab report. You do want to put your lab reports, your data in your lab reports in tables, okay? Because tables are nice and neat, all right? Okay, you also want to have a control. In a lot of experiments, they do set up a scientific control, all right? That way it's used to basically compare something to, all right? Okay, scientific control, an experiment set observation designed to minimize the effects of variables other than the independent variable. This increases the reliability of the results, often through a comparison between control measurements and other measurements. Okay. Okay, so once you're done with the data section, all right, you have a analysis section, all right? And the end, you guys will have your analysis section. Okay, so in your analysis section, you write down all the patterns and trends you see in your data. And you always refer back, okay? Because the data, the tables, that's the evidence that you will use, okay? And you'll always cite your sources, whether it's from your own data or whether it's from another person's data, okay? You always cite your sources, okay? So in this case, this is a paragraph. You basically kind of write down what trends you see, okay? For example, if you see like the bubbles using the sucrose was larger consistently, or if the bubbles using the sodium chloride were smaller, whatever the case. All right, so this is basically again your data section emphasized and put into a paragraph. Okay, that's your analysis section. Okay, and again, the analysis, all right, you will write about what happened in the ANOVA, what happened in the chi-square, what happened in the t-test. For example, according to the ANOVA table, figure 1, it seems that there is significant difference between the control bubble solution, the sucrose bubble solution, and the sodium chloride bubble solution. All right, that's something I would have in my analysis section. All right, for the ANOVA. And remember, this part of the lab report, it has to be a paragraph. All right, or multiple paragraphs. Okay, lastly, the conclusion section. Okay, your first statement will be whether your null hypothesis or your alternate hypothesis was accepted. Okay. Secondly, how does your data support your accepted hypothesis? Okay, now this should be a pretty large paragraph here. All right, explaining how your data supports. Okay, come up with inferences. All right, compare to similar studies. Okay, there should be a lot of collaborators in similar studies. Okay, whether it's looked up or whether it's a co-collaborator that's from like another class or something. Okay, and then lastly, you just want to finish with what future studies you want to do. All right. Now these three things should all relate. Okay, so what your future plans and your future studies will be, it should relate to how your data supported your hypothesis or how your data supported your null hypothesis. Okay, was there something wrong with the research? Was there something wrong with the experimental design? How will you fix that? Okay, if the alternate was supported, okay, what else related problems can you solve? How can you refine your hypothesis to further solve more problems? All right. And that's how you document science in the proper lab report format. If you like the video, like, subscribe, and just kidding. Just post a comment if you guys have any questions. I'm sure you guys have a lot. And bear with me, this is like my second time doing this.